Hello, my name is Annabelle Lingbeck, and this is my project called The Effect of AKT2 on Astrogliosis. So to start, we first have to understand astrocytes and their importance. Astrocytes are special cells that are found all throughout the central nervous system, which includes the brain and the spinal cord. Astrocytes are called astrocytes because they look like stars. As you can see in picture 2, they have a lot of spindly edges and arms that make them very complex cells. They are important because they surround neurons and provide insulation, which means they are very closely correlated to memory and brain function. So my project focuses on astrogliosis, which is a process in which astrocytes undergo changes to their structure as a result of CNS insults or events. These events are traumatic things that could happen to these cells that would cause them to split uncontrollably and also get more complicated. So in astrogliosis, there will be more astrocytes present and they will be too complicated to do their own normal processes. Astrogliosis is related very closely to neuronal conditions like Alzheimer's, which is why we are doing this research, is we want to see if there is any way we can make better the effects of astrogliosis. So what I looked at for this was AKT. AKT is a signaling pathway that regulates and affects many parts of the cell. There are 3 different types of AKT, AKT 1, 2, and 3. My project specifically focuses on AKT 2 because AKT 1 and 3 have been widely researched and there was a connection found between them and astrogliosis. So my project is focused on if there is a connection between AKT 2 and astrogliosis. So my question is if AKT2 has an effect on astrogliosis, and my hypothesis was that it would. I predicted that AKT2 would regulate astrogliosis and make the effects lesser, hopefully even slow down or stop the process of astrogliosis. So this research is significant because a connection between AKT2 and astrogliosis would mean potential ways to treat several neuronal diseases like Alzheimer's with therapies and such. So I am using brain tissue samples from 4 different groups of mice to measure the presence and density of the astrocytes in each of these groups. So for my procedure, I used brain samples from 2 groups of mice, wild-type mice and knockout mice. Wild-type mice have AKT2 present in their central nervous system, while the mice with the allele knockout do not have AKT2 present. Each mouse is treated with either LPS, which is lipopolysaccharide, an endotoxin that induces astrogliosis, or saline, which is used as a control. And these treatments happen 24 hours before the mice are sacrificed. So in my procedure, I have 4 different groups of mice: a wild-type mouse with LPS treatment, a wild-type mouse with saline treatment, a knockout mouse with LPS treatment, and a knockout mouse with saline treatment. So the procedure is focused mostly on the process of a Western analysis, which is done by older, more experienced members of the lab, since it works closely with brain tissue and also a lot of harmful chemicals. In a Western analysis, proteins are separated out and then stained so we can visualize different parts of the brain tissue sample. The visualization is blots of each of the proteins on pieces of paper. For my project, I obtained the images from the Western analysis and used ImageQuant data processing site to produce numbers and graphs that represented the density and also the presence of astrogliosis. So this is what a Western analysis plot is going to look like, and there's a lot of different aspects of it that we need to go through to understand what's going on. This top bar up here, this is going to be which mouse the sample came from. So this first all the way down this column is a saline mouse. The second one is an LPS mouse, all the way down the column, saline, LPS, and it goes like that all the way across. This second bar right here shows that if there is AKT2 present in the sample, so this is going to tell us which of the mice are wild type and which of them are knockout. In this case, these first two are wild type mice, and then these two over here are also wild type mice, and then all the way down the last two are as well. We can tell because they have darker bands of AKT2, they're more dense, which means they have it present. The rest of these mice then are knockout. This last band down here is an indication that the test is working, that the Western was done correctly, because each mouse has this GAP-DH present in their protein samples, and all of the bands should be relatively the same size. This third band is the one that I was most concerned with, because this band called GFAP is a representation of the amount of astrocytes present in the protein. So for my project, I wanted to see variation between each of the bands, representing whether or not astrogliosis was occurring more frequently in each type of mouse. So along with these blots, I was creating graphs for them. This first graph is a representation of the total AKT2 present. So all of the wild type mice should have these big bars, while the knockout mice should have small bars. And also, for all of the graphs, the white bars are representing saline, and the black bars are representing LPS. So we can tell that the Western analysis worked because my graphs are showing a really large difference between the total AKT2, since knockout mice do not have it present. The second graph, the measure of GFAP, is the one that I was most concerned with again. It should show variation between the amount of astrocytes present, and we can see this variation because astrogliosis causes more astrocytes to be present in the protein, which means that the bars with the most astrogliosis should be bigger than the bars without as much astrogliosis. So for my prediction, I wanted to see that the wild type bars were showing significantly less astrogliosis occurring than the knockout bars, which did not have AKT2 present, because my prediction was that AKT2 would regulate the astrogliosis. As you can see from this graph, they are all very similar. Each of the bars there's no discernible difference between any of them, which means that my prediction was wrong, and I proved my null hypothesis that there is no correlation between the presence of AKT2 and astrogliosis. So for the future, there are plenty of things to be doing since there were limitations with my study. The biggest one that I would like to test more is that the LPS was only given 24 hours to take effect in the mice before they were sacrificed. This might not have been long enough to see if the astrogliosis would occur. If they were given longer time, like potentially for 48 or 72 hours, there would be more astrogliosis, and we could be able to tell the difference between each of the protein samples a bit more. There was also a lot of room for error in my data, both in the Western analysis and in the use of image quant. Because Western analysis is a complicated process with so many steps, there's a lot of things that could go wrong in any one of those steps. If there was a misstep, could have caused all of my data to be incorrect. There could also have been incorrect use of image quant on my end of things. If I incorrectly measured the densities of the proteins, or if I told the machine to do something that I wasn't supposed to tell it to do, it could have messed up my data and caused it to be incorrect. This data is really important to me because I am really interested in learning more about what causes Alzheimer's and many other memory disorders, and I'm really excited to continue in the future with this research because it is needed to see if there could potentially be a connection between this and hopefully be able to develop new therapies and ways to treat neuronal conditions. And this is my bibliography. Thank you so much for listening.